Okay, guys, you're welcome to Nisiki Academia. In this video, we are going to be looking at SLT 103.2 laboratory techniques, and we'll be focusing primarily on basic exam focus and, of course, major area of concentrations. All right, in the course. Okay, so I'm going to be running through the area of concentration I was able to put together. All right, the major area of concentration as far as this course is concerned. Okay, and you have to understand that this is a laboratory course, of course, laboratory techniques. All right, so of course, you have to understand laboratory design. We study with laboratory design, safety, and equipment. Look at everything concerning laboratory structure. Okay, if you watch my previous video on 350 questions that I put together and the entire summary of the course, you would be able to realize how the design of the laboratory is supposed to be, how the lighting is supposed to be, how the ventilation and, of course, temperature control. Okay, then the next one is safety features. All right, safety features, of course, when you start looking at fire extinguisher, first aid kits, aprons, goggles, gloves. Okay, then also have a very good knowledge of handling of gas. Okay, handling of gas, electricity, and water in the laboratory. Okay, then also know how chemicals and reagents are properly stored. So study the proper storage of chemicals and reagents. Then, of course, the approach. I actually had to put the approach together. The approach that you actually use in memorizing this, memorize the safety rules. Okay, draw diagrams of lab layout and identify equipment and uses. Or if you don't want to necessarily draw the diagrams of lab layout, okay, go online and look at the standard way of actually setting a science laboratory and identify all the equipment there, and of course study their uses, the uses of different equipment. All right, then the next thing is laboratory glassware, plastics, and materials. Before we continue, if you've not subscribed to this channel, please do well to hit the subscribe button so you can get notification of previous videos. Okay, and of course, if you find this video beneficial, do well to share and, of course, like as well. So laboratory glassware, plastics, and materials. Now, of course, we have to look at the types of glass. Okay, we have borosilicate, soda lime, and pyrex. Look at the differences between this borosilicate glass, soda lime glass. Okay, and check what Pyrex is all about. Okay, you can make reference to that video as well, and of course you can also send a DM for the complete PDF of that containing all the 350 revision questions exercises in the course. Okay, also look at types of plastics. We have the PVC, which is called the polyvinyl chloride. Okay, we have the polystyrene, we have the polypropylene. Look at how they are actually applied, more like the industrial application and laboratory application of these plastics. Look at the uses, advantages, and disadvantages of each of them as well. Then the approach to this is create a comparison table between the PVC, polystyrene, and polypropylene. Okay, then match equipment with the correct materials. Those equipments that you are able to identify here, those equipments you are able to identify here, come here and match the equipment with the kind of materials that are used in producing them. Are they produced using PVC or polystyrene or polypropylene? All right, so do that, match equipment with the correct materials. All right, so the next thing, of course, is microscopy and microscopes. All right, so of course you have to understand um the entire meaning of microscopy, the entire concept of microscopy. Okay, I also put everything together in that PDF containing 350 revision questions. I also put the answers there. Okay, so microscopy, and of course the different types of microscopes. Okay, look at the different parts: the ocular lens, the stage, the condenser, the diaphragm. Okay, the nose piece and all of that. There are many different parts of the microscope. All right, then look at the types of microscope: the simple, the compound, the electron microscope. Okay, the light field. There are many types of microscope. Then look at the history. Okay, look at how Robert Hooke, how Leeuwenhoek and Entruska, okay, how they contributed to the development of microscopes. Now, the concepts, look at what magnification is, all right, look at what resolving power is, refraction is as well, and of course, you have to know their formulas. Now, the best approach to this is label microscope diagrams, okay? Check diagrams of of microscope and look at their parts, their labeled part. Now, practice magnification calculations. Look at calculations that have to do with you using the formula of magnification. Okay. Then of course use historical timelines. What do I mean by using historical timelines? I mean that you should have the understanding of the different dates that each of these guys contributed to the development of the microscope. Robert Hooke, Ewan Hooke, and Ernst Ruska, the different years, different dates, and all of that. Those are historical timelines. They also have aseptic techniques. All right, aseptic technique. You know what aseptic techniques are now? The techniques that are actually employed in the laboratory to avoid any form of introduction of bacteria, fungi, and of course any form of microorganism that can lead to the disorderliness and degradation of the laboratory. All right, so look at aseptic techniques and microbial culture. We have the inoculation, incubation, inspection, isolation, identification. All right, understand what each of these mean. All right, the five eyes. Then now use of inoculating loop, flaming streak plate, and spread plate. Understand their different uses. Okay, then also check how to prepare a smear and how to fix heat, also known as heat fixing. Now the approach to this segment is to write the step-by-step procedures. All right, step-by-step procedures for the aseptic techniques. All right, then draw diagrams. Now the diagrams you should draw are of course the different diagrams that are involved in inoculation, incubation. If you don't have all the time, also check up the diagrams online. All right, that involves the five eyes. All right, check up the diagrams that involves inoculation, incubation, inspection, isolation, and identification. All right, then of course understand contamination prevention, how to prevent contamination. Of course, that is the general idea of aseptic techniques. Okay, the steps that can actually be taken or employed in preventing any form of contamination. All right, so that is another one. Okay, now we have culture media and microbial growth. Okay, so you need to know what the types of culture media are. They defined the complex, selective, differential, enriched media, okay, and examples of each of these media. We have agar versus gelatin. Check the difference between agar and gelatin. Understand what blood agar is, where it is used, how it is cultured. All right, the McConkey agar, the potato dextrose agar. Now also understand the classification. You know, you have the liquid, the semi-solid, and of course the solid media among the different types of classification. Then the approach here is create charts with examples. Now, what charts should you create? Create charts of of these different types of media. All right, create charts and write the examples of each of them. Then memorize media bacteria pairings. All right, media bacteria, that is of course the type of media and the example of bacteria that can be cultured in that media. All right, so that's what we call media bacteria pairings. Then practice the classifications. All right, then the next thing is colony morphology and identification. Understand the difference between shape, elevation, margin, size, and pigmentation. Okay, understand that. Then of course colony types, secular, irregular, and filamentous. Okay, so check the difference between these. Then the best approach to this is to draw the colony types and memorize the descriptive terms. Okay, the descriptive terms, that is of course the terms that are used in describing the secular, the irregular, and the filamentous of course colonies. All right, then of course then the next aspect of it is plant tissue preservation and processing. All right, herbarium techniques. Okay, so you study herbarium techniques, but the methods now, of course, you have to understand the methods are involved in drying. Okay, drying a particular tissue, how to fix a particular tissue, that is fixation. Then we also have the cryopreservation, freeze drying, and of course embedding. Then of course tissue processing. All of these things are in the video I actually made on the revision questions involved in the course. Okay, laboratory techniques. Okay, then of course study tissue processing, the understand what dehydration is, clearing, and of course understand the concept of embedding when it comes to tissue processing. Then you need to also know what a microtome is, the different types of microtome, what it functions as. Okay, understand what a cryostat is, then of course know the methods that are involved in staining. Now the best approach to this is that study the preservation methods with examples. Okay, that is the best approach to remembering. Study preservation methods with examples, label diagrams, and understand tissue sectioning. All right, so that is that. Now specialized lab equipment. Okay, this one is actually you studying equipment, special laboratory equipment in the laboratory. Okay, understand what the hot plate is. Okay, understand the principle of operation, understand how it is used. All right, know everything about centrifuge, autoclave, and different balances, weighing balances, and all forms of balances. Now, understand the principles of autoclaving, having a temperature of 121 degrees Celsius, 15 psi, and of course, operating between an effective range of 15 to 20 minutes. Okay, then of course, understand the centrifuge balancing rules. Okay, how to balance a centrifuge. Okay, you know, a centrifuge, of course, is actually used in the laboratory. Okay, is for centrifugation. Okay, when you're spinning samples, all right, that's centrifugation. Now the approach now is to write notes on the principles and memorize numerical values, especially these values. This one is just an example for under principles of autoclaving. All right, for principles of centrifuge you also have there, or hot plates you also have that. All right, then of course you need to understand the different numerical values, constant values that are attached. All right, then the general study tips that I was able to put together, okay, that will help you. Group similar topics for easier recall. I've been saying this all the time. Okay, topics that are similar together. Okay, group them together, especially when it has to do with fixation techniques, and of course embedding and all of that. Okay, and of course sectioning. Okay, those topics that are similar in nature, group them together, then practice filling blanks. Okay, since many questions are structured that way, so I was like, use my 350 questions bank. All right, use my 350 questions bank, then use mnemonics. Okay, use mnemonics for easy recall. For instance, the five eyes. Okay, the five eyes when it comes to laboratory techniques. All right, is one mnemonics that can actually help you to remember. Mnemonics are more like abbreviations. All right, abbreviations that can easily help you. Okay, for instance, now if you want to look at um if you want to look at major specialized equipment. Okay, major specialized equipment, you can say autoclave. We have the autoclave, we have the centrifuge. Okay, we have the hot plate. Okay, we have the hot plate. So you can say ash, autoclave, centrifuge, hot plate. Okay, that just is little example, but use abbreviations, okay, to help you to remember and of course to easily recall. Now focus on definitions very well, examples and procedures. Know how to define different terms. Focus on it very well. Focus on examples of different things. Remember, we talked about, of course, media, classification of media, and the different examples of bacteria that can actually be cultured in that media and all of that. Okay, understand examples. You should know the examples of virtually every concept in this course, of course, and of course, know different procedures. Okay, we talked about aseptic techniques, we talked about herbarium techniques, we talked about culturing and all of that. Okay, these are different techniques. Okay, inoculation has its own processes and procedures as well. Identification has its own procedures. Isolation has its own procedures. Okay, so no procedure. Then dedicate time to diagrams. That is microscope, lab setup, and colony shapes. So you can be able to differentiate. Okay, can be able to differentiate um the different types of colonies. Okay, using their different shapes. All right, dedicate your time to study microscope so you can be able to know the different parts of a microscope. You don't even need much time to look at lab setup, because you need to basically know how lab is structured, how it is painted, how it is ventilated, how the illumination, the lighting, and all of that is like. Okay, so that's it basically. All right, so I believe that this is going to help you. Okay, this is basically the major, okay, area of concentration as it regards this course, SLT 103.2. If you found this video helpful, do well to share. And of course, if you have any question whatsoever, write a question in the comment box below. If you've not subscribed, please do well to subscribe so you can get notifications of our subsequent videos. Thank you and good luck in all your adventures in science laboratory technology, laboratory techniques. Bye bye.